Hi guys, we're continuing. This is cell biology and life science in a nutshell. We're going over my notes. We're discussing cell division. Here we have a cell shown. So cell division occurs whereby you have chromosomes that are duplicated. So a chromosome consists of two strands of DNA, which are called sister chromatids. And in cell division, each one of the sister chromatids, they line up in the center of the cell in this stage called metaphase, and with the help of this spindle fibers, which are fibers, protein fibers that help with controlling of the movement within the cell. So you have asters, which we learned about earlier, and which are part of the whole process, and the asters help with pulling apart of the chromosomes from the middle, so all the genetic material gets pulled apart and put into two separate nuclei, and in the final phase of cell division, two separate cells are formed because their cytoplasms split as well, so they become two separate cells with the genetic material dividing between them. So the spindle apparatus and the asters, which are at the ends, pull apart these genetic material. And this is showing another view of the asters, what the fibers are looking like, and the spindle apparatus as well, with the chromosomes in green and the fibers in purple. So this is the process as far as the as far as the physical division of the cells goes, and so let's see. Also, mitosis and meiosis use these fibers for helping of the division of the cell, and okay, so mitosis involves occurs in both prokaryotic and eukaryotic cells. Meiosis occurs only in eukaryotic plant cells and fungi. So okay, in mitosis is the stage of the cell cycle that involves division of the cell nucleus and separation of chromosomes. So mitosis results in two daughter cells, and meiosis, in contrast, results in four daughter cells. So we'll get into that. But for now, for mitosis, you use for cells in the somatic cells mostly of the body, for multiplication of cells. So after cytokinesis, when the cytoplasm splits of the one cell into two separate cells, so then the two distinct cells are formed. And these are showing all the steps. So prior to mitosis, the cell prepares for division by replicating its DNA, increasing its mass and the organelle numbers, and getting ready to divide. Chromosome movement also occurs in the different phases, helped by the fibers we discussed earlier. And the stages, as shown here: prophase, metaphase, anaphase, telophase. So in interphase, before the division begins, the cell undergoes the period of cell growth. So it says some 90% of a cell's time in the normal cellular cycle may be spent in interphase. So presumably 10% is in the division cycle, where the cell begins to divide into more cells, etc. So step by step, the G1 phase, the period prior to synthesis of the DNA. In this phase, the cell increases in mass in preparation for cell division. The G1 stands for gap. So gap 1 is the first gap phase. Then the S phase, the period during which DNA is synthesized. In most cells, there is a narrow window of time during which DNA is synthesized. So S means synthesis. G2 phase, period after DNA synthesis has occurred, but prior to start of prophase, the cell synthesizes proteins, continuing to increase in size. G in G2 also gap, so second gap phase. Then in the latter part of interphase, the cell still has nucleoli. The nucleus is bound by nuclear envelope normally, and after synthesis, the cell's chromosomes have duplicated, but they're in the form of chromatin. In animal cells, two pairs of centrioles formed from the replication of one pair are located outside of the nucleus. So as we saw, centrioles are fibers, and they help. They're part of the cytoskeleton, so they help with positioning of the controlling how the nucleus and the chromosomes are going to be moved and pulled apart. That's showing the cytoplasm during the time of the cell pre-cell division or after. Here the cells dividing at the final stage. The asters pulling apart the chromosomes. These are the asters as well, the fibers, and they're pulling apart one cell into two. These are the nuclei and the spindle fibers and the centrioles. Yeah, so here's another pic of centrioles, what they look like. They're like cylinders. So they're near the nuclei, and these fibers help with pulling apart of genetic material in the nuclei. So early prophase, chromatin fibers become coiled into chromosomes, with each chromosome having two chromatids joined at a centromere. So the centromere is consists of the centrioles. It's a unit, fiber unit. So the mitotic spindle, composed of microtubules and proteins, forms in the cytoplasm. In animal cells, the mitotic spindle appears as a structure called asters surrounding each centriole pair. The two pairs of centrioles formed from the replication of one pair in interphase move away from another toward opposite ends of the cell due to the lengthening of the microtubules that forms between them. We'll continue in the next part.